Hello friends, I am here with my channel Solving Questions with Gauri ma'am. Today I am going to explain one question from IIT JAM Biotechnology entrance examination. Western blot is used for the identification of options are monosaccharides, RNA, DNA, or whether it is a protein. So going option wise, discussing first option. First is a identification of monosaccharides. So when we need to identify monosaccharide, we use Barfold's test. Here there is a copper acetate get converted into copper oxide and this gives a brick red precipitate. So this is the test used for the identification of monosaccharide and hence this is not the answer. Some other tests like Molish test, Benedict's test, Selimov's test, these are used for further identifications like it is a hexose sugar or whether it is a aldehydic sugar or ketone sugar. It is a pentose sugar. So further identification can be done with another test. One of them is a Fairling's test, but Western blot is not used for the identification of monosaccharides. So moving to next option. Yes, the second option is given as a RNA. So for the identification of RNA, we use northern blot technique. What do we do in here in this technique? First of all, RNA are separated by gel electrophoresis. Then the bands are transferred onto a suitable membrane, preferably nylon membrane is used here. It is immobilized on the nylon membrane. Then these are hybridized with a radioactive single-stranded DNA probe. Further, how do we detect? Band showing hybridization are detected by autoradiography. So this is in short, I have explained the procedure for the northern blot. So let's see what is the Western blot used for. So moving further towards the option C. Option C is asked for the DNA identification. So for the identification of DNA, we use Southern blotting technique. What do we do here? First, genomic DNA is taken. It is carried out with a restriction endonuclease digestion. So it will give number of bands. Now these fragments, DNA fragments, are run on a agar gel electrophoresis for separation. Then alkali treatment is given here for the separation of DNA double strand. Now on a membrane we get single-stranded DNA. On a gel we get single-stranded DNA and these single-stranded DNA are now transferred to the nitrocellulose filter. Further, these are hybridized with a single-stranded radioactive probe. Probe means it's a DNA only. So it is hybridized with a single-stranded DNA and using the autoradiography we can identify DNA in a sample. So in this way this southern blotting technique is carried out. Southern blotting for DNA, northern blotting for RNA. Now let's see what is Western blotting used for. Coming to the last option, option D is for the protein identification. And yes, this is the answer for the question. For the identification of protein, Western blotting is used. So during the Western blotting, what is the procedure? Protein is first carried out with a polyacrylamide gel electrophoresis which separates the protein bands. And now these are transferred to either a nitrocellulose membrane or it may be a nylon membrane. Further, these are detected by using the antibodies or it can be a lectin. these can be hybridized, and if it get hybridized, then we come to know the protein is present. So this is the technique used for the protein identification. We can summarize like this: for DNA identification, southern blotting; for RNA identification, northern blotting; and for protein identification, we use western blotting technique. In case of DNA, the hybridization is done with a complementary DNA probe. In case of RNA, again the DNA probe is used, so it forms a RNA DNA hybrid. And in case of protein, it is detected by hybridization with antibodies or lectin. So when we say about membranes, for the identification of DNA, the transfer is done on a nitrocellulose membrane. In case of RNA, it is done on a nylon membrane, and for protein, any of the membrane can be used. So these are some differences. The basic procedure for the technique is similar. Here I have concluded with today's video. Keep watching my videos. The exam is 14 days left for your exam. So all the best for your exam. And I request you to please like, share and subscribe for my channel, and also to hit the bell icon. Thanks for watching.